This is our journey, step 1, into the porous organisms, the sponges. Here is our taxonomy. You can see that we are again broken down into two phyla, phylum Calcarea and phylum Silicea. Phylum Calcarea has two classes underneath it, Calcarea, Demospongia. Phylum Silicea just has a single class under it, Hexactinella. So what if you're a scuba diver? What if you're diving around? Well, the first thing to know if you were going to identify a sponge is it's probably something that looks like it has a bunch of holes in it. Okay, so you're diving down and you see something that is, let's start from the bottom up, very glass-like. It looks like it has holes in it and it's glassy. Well, that's going to be class Hexactinella. So you're a scuba diver, you're diving down, you see something with a bunch of holes in it. It's not glassy, but it looks big, and it looks like something you might be able to wash your car with. That's going to be class Demospongia. So you're diving down, you're a scuba diver, and you're looking around and you find something that looks like it might have holes in it. It's not glassy and it's not big, and it doesn't look like it's something you can wash your car with. And you have a little microscope with you or a hand lens, and it's probably going to be class Calcarea. So that can help you out not only with if you're scuba diving and looking for sponges, also on a lab practical. The evolutionary significance of sponges is they are the base of the animal kingdom, and DNA evidence supports this measure. They are known for having only the cellular level of organization. They're the only animals that have just the cellular level of organization, which means that sponges truly do not have tissues. Also, we see a descent with modification application here, in which we have three main body plans, and we'll look at these three, the asconoid, the cycanoid, and the leuconoid, and we'll look at all three and how they obviously display descent with modification. Here is some of the anatomical features that we're going to look at in the sponges, and if this seems a little overwhelming, just go ahead and start taking a few notes, and we'll look at each one of these in the subsequent slides. But they are suspension feeders, but they actively pump water through them. One of the ways they do that, they have special cells with flagella that whip up a current, and we'll even include a video in this lecture where you can see them pumping. Again, it's part of their evolutionary significance. Their body is made up of cells as functioning as a unit, but no true tissues. And again, they're the only animals that do this. The internal anatomy of a sponge, it has a central cavity to it called a spongiocele, and that spongiocele can vary depending on its body type. But that spongiocele is where we pump all the water into, and then eventually water exits through an opening we call an osculum. The methodology of pumping the water is done by a special cell called a choanocyte. They are flagellated, and that flagellated whips around, and it pumps that water through the sponge, through these incurrent canals, into the spongiocele and out through the osculum. The choanocyte also features a basket on it called the microvilli that helps it catch and recruit food particles. The other cell type that we're going to look at on the sponge is the amoebocyte, and they're responsible for nutrient delivery and the creation of structures. So if the choanocyte is pumping and catching food, the amoebocyte takes care of everything else. Sponges are hermaphroditic, which means they have both male and female parts. We will see on most of the basal animals that we do have hermaphroditic conditions. As I mentioned before, there's three basic body plans. The most simple, asconoid, the second most simple, the cycanoid, and then the more complex leuconoid. Their bodies produce two structures that help them with structure and support, spongin and spicules. Spongin is proteinaceous and is composed pretty much just like human hair, and that kind of keeps everything together. And then the spicules can also add for rigidity and a structure, and they can also ask for a little bit of defense. And they're typically made out of a calcium carbonate or a chalk. And you'll see that they have a very distinct geometry to them. Here is a basic idealized sponge anatomy. Water flows into the incurrent canals, into this central canal we call the spongiocele, and then out through the top opening, which is the osculum. So again, the water flows in through the incurrent canals, into the central spongiocele, and then out through the osculum. The mechanism for getting the water to flow is again through this choanocyte. And you can see here these choanocytes, these purple cells, are flagellated and they whip around and create that current. You also have that basket called the microvilli to catch the food. You have other cells here, the amoebocyte, and they're responsible again for day-to-day functioning and nutrient delivery. And you can also see spicules, these geometric shapes here that are adding rigidity and a little bit of protection. So that's basic sponge anatomy. Here's a closer look at the two cell types I want you to know. The choanocyte, again, it's flagellated, and that flagellated cell will whip, 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 and create a current. That current will help catch food through this basket we call the microvilli. Food particles are then shuffled over to the amoebocyte for distribution and to figure out what to go ahead and to do with the rest of the structures. Here's a good look at the choanocytes as they line the internal structure of a sponge. And here is a cross section, and you can see that the inside of this sponge, what you're looking at right here down at the bottom, is the spongiocele, and you have a ton of these choanocytes lining the inside of the canals. So let's look at the three different body plans, the asconoid, the cycanoid, and the leuconoid. The asconoid is the simplest of these. A good way to remember that is the letter A starts the alphabet. So the letter A in asconoid starts sponge diversity. The way that we look at each one of these is the levels or layers of choanocytes. In the asconoid body plan, the choanocytes line a singular level of the sponge. Water flows inside, being pumped into the spongiocele and out through the osculum. Again, a single layer of choanocytes around the inside of the sponge. Class Calcarea is a good example of an asconoid. All of the class Calcarea, the class Calcarea are going to end up either asconoid or cycanoid sponges. Here is the cycanoid, again, so you probably have class Calcarea. Water moves in through the incurrent canals again, but you can see we have two levels. So cycanoid has two levels of choanocytes lining a double layer, and that allows for extra water flow to be pumped by two layers of choanocytes, an increased surface area, and you also have a little bit of a current, and as that current moves around, it can slow water down and slow food particles down. As a result, because they can recruit more food, they grow larger. But water still flows in the incurrent canals through the help of the choanocytes into the spongiocele and out to the osculum. This is a real good look at descent with modification. And again, class Calcarea is comprised of the asconoid and the cycanoid body plans. This is the most advanced plan, the leuconoid. The leuconoid are going to be all in class Demospongia. They grow larger, and the reason they can grow larger is we have a more complex network of choanocytes. And you've got all sorts of currents that can flow, spin around, thanks to the multiple layers of choanocytes. But water still flows into the incurrent canal, into the spongiocele, and out through the osculum. So it's the same basic design, but what you end up with is a more complex structure. The more complex structure has greater surface area. You have greater surface area. It in turn allows you a chance to recruit more food particles. You recruit more food particles, you can grow larger. So these are the Demospongia, or the larger sponges. And here's a good example of the Demospongia. You can see they're much larger. Class Demospongia. So how do you apply this? Well, we sort of already talked about it, but we know that the phylum Calcarea is broken down into two classes, class Calcarea or Demospongia. all of class Calcarea is either asconoid or cycanoid. All of class Demospongia is leuconoid. So when all else fails, you can just look at the structure and say, well, if it's complex or it's large, chances are I've got a Demospongia. Now, we haven't talked yet about phylum Silicea, the class Hexactinella. Those are sort of a modified asconoid. You're going to see the pictures of these glass sponges, and they're going to have a very simple body plan. Internal sponge anatomy, again, we have two structures that make that up. It's either the proteinaceous spongin, which sort of connects everything together, not in a tissue, but connects everything together, and then the spicules, and they add support and protection. When you look at the spicules, you'll see that their calcium carbonate structure sort of has a pointy geometry, and that pointy geometry would make them unpalatable to many would-be predators. Here's the spongin. And here's the spicules. Here's a bonus image, spongin and spicules together. Sponge reproduction, being the basal animals, it's not a real advanced form of reproduction. Their sexual reproduction is done by something called the smoking sea, in which they eject sperm into the water column and hope that it lands on a surrounding sponge. It's a very massive effort with a low success rate. They can also reproduce asexually, either through an asexual process that's sort of purposeful or actually through budding off. They could create a little bud structure and just sort of pinch off. Since they only exhibit the cellular level of organization, you can in fact take a sponge and grate it up and you'll end up with more sponge. So again, this is Kingdom Animalia. These are the sponges, and here are the two phyla that we're going to look at, Calcarea and Silicea. The claim to fame for class Calcarea under phylum Calcarea is that they are the asconoid or the cycanoids, and we note that those are the most simple of the body plans and represented by the lab model, which we'll look at on the next slide. Here is the idealized sponge model. We know water comes in through these in-current canals into the spongiceal, and then out through the osculum. And here is complete sponge taxonomy. Class Demospongia, the claim to fame here is those are all leuconoid sponge. So they are the more complex of the body forms. And conversely, they allow to get bigger. You will find your commercial sponges in here. And if you're a scuba diver, you might ask, can I wash my car with this? Here's a couple looks at some Demospongia. And lastly, in the diversity of sponges, class Hexactinella. We haven't seen this one yet. This is sort of a modified asconoid in that it looks and appears simple, but it's modified. It doesn't have that idealized shape. They do make a mutualistic relationship with some small shrimp, and that story is why they're given as a wedding gift in some Pacific cultures. The small shrimp actually become trapped inside the euplecta, and they live their entire life there together. So it's sort of an analogy to for marriage. It's a mutualistic relationship in that the sponge, the glassy sponge, protects the invertebrates, and then they're messy eaters, so the sponge can get a little bit of extra food there. Here's another look. Sponges are good and bad for the environment. They can be good for ecotourism and zooplankton and contribute some of the good microorganisms of the ocean. They can also be invasive. In the bottom right here we have a picture of an encrusting sponge that's actually taking down a coral head. So they're good and they're bad. I'm going to click out here and check out a sponge video, and it's really cool, and it shows you the filtration and how they pump the water in through the choanocytes with the whip, and it really shows that they're actively pumping seawater for filtration of food. So We have just finished the poriferans, and that is the non-true tissue organism. Now we need to begin our journey of the true tissue organisms, and our first stop off here is going to be in the radiata, and the radiata is going to be the cnidarians and the ctenophorans. So that's next.